The next section of this unit will be on pathogens and our immune system. Our immune system is the body system that's made up of special cells, tissues, and organs that function in fighting disease. Our immune system recognizes invaders, attacks those invaders, destroys those invaders, and then remembers those invaders. A disease is a condition other than injury which disrupts normal body function. There are lots of different kinds of disease. There are genetic diseases. There are deficiency diseases, such as iron deficiency. There's physiological diseases, and then there are infectious diseases. And the infectious diseases are the ones that we're going to be focusing on with our immune system. An infectious disease is an infection by a pathogen, and a pathogen is a disease-causing agent. Examples of pathogens: fungi, protists, animals or parasites, bacteria, and viruses. Disease can disrupt normal body functioning by destroying cells or damaging tissues, releasing toxins or poisons, and disrupting cell functions. Virulence is the strength of a pathogen. It's the pathogen's ability to cause damage to its host organism. So low virulence would mean that the pathogen is not very strong or does not cause much damage to the host, where a pathogen with high virulence would be much stronger and can cause a lot of damage to the host. Here are some examples of pathogens and the diseases that they are related to. Of all the pathogens, we are going to focus mainly on bacteria and viruses. Bacteria are prokaryotic cells, and in terms of bacteria, most are non-harmful or actually beneficial. For example, we have bacteria which live in our intestines that are essential for us to be able to properly digest food. There's bacteria that's used in food. For example, yogurt relies upon bacteria and decomposers. Without decomposers, dead organisms would just pile up. Very few bacteria are actually pathogenic, but if they are pathogenic, they will often break down an organism's tissues and use as nutrients, and they also often release toxins. Here is a list of the 14 deadliest bacterial diseases. And when you look at the slideshow on your own, if you're interested in any of those diseases, if you click on it, it'll actually take you to a web page that has more detailed information on it. Other common bacterial diseases: food poisoning, as well as salmonella, which is a type of food poisoning, ear infection, urinary tract infection, staph infection, strep, and chlamydia. All of those are bacterial infections. Here are some examples of different kinds of bacteria. Notice that there are different shapes and appearances of different bacteria types. Bacteria can be treated with drugs that are antibiotics. Antibiotics are medications that only work on bacterial infections. Antibiotics do not work on any other pathogen. What antibiotics do is that they destroy the bacteria or slow the growth. And by slowing the growth, it also then allows our immune system to be able to fight it off better. There's many different kinds of antibiotics with different strengths. There's penicillin, amoxicillin, cephalosporin, erythromycin, And so depending on the type of infection and the severity of the infection, that will help the doctor determine what kind of antibiotics to prescribe. Here is a comparison of bacterial growth with penicillin and without penicillin. And so you can see that the penicillin is actually destroying the bacteria, which reduces the amount of reproduction they're able to do. So which antibiotic should be used for bacteria? And this is often determined through a Kirby-Bauer test, which tests the effectiveness of various types of antibiotics. And so bacteria is spread in a petri dish, and then discs with different antibiotics are placed on that petri dish. After allowing the bacteria to grow for a certain amount of time, the scientists will look for something called the zone of inhibition. The zone of inhibition is the area where there's no bacterial growth, and it indicates the antibiotic's effectiveness. So wherever you would see a larger zone of inhibition, that worked the best at stopping the bacteria from growing. So on this slide, we can see that A was not effective against this bacteria, and C was the most effective as it had the largest zone of inhibition. So on the slide, the order of the effectiveness would be C, B, A. The bigger the zone of inhibition, the more effective the drug was. And here's an example. Now, over or inappropriate use of antibiotics has resulted in bacterial resistance. And what that means is that certain antibiotics are no longer effective against certain bacteria strains. This occurs through natural selection. Weaker bacteria are going to be killed first by the antibiotic. The stronger bacteria are able to survive and pass that resistance on to their offspring. So if you are supposed to take antibiotics for a certain length of time and you stop taking them before the prescription runs out, you may have killed all the weak bacteria, but you were not taking the antibiotic long enough to kill the stronger bacteria. And those stronger bacteria then survive. They reproduce stronger bacteria just like themselves. And then you could be passing that stronger strain of the infection on to another individual. Here's an example in which some E. coli bacteria have sacrificed themselves and absorbed large amounts of the antibiotic so other cells are able to survive. The bright green bacteria on this slide are absorbing large amounts of the antibiotic, sacrificing themselves, knowing that they're going to die, but other cells will be able to survive and reproduce. So they're certain cells that are basically protecting other cells. So now moving on to viruses. Viruses are the other pathogen that we are most often infected by. Now, unlike bacteria, all viruses are pathogenic and all organisms are susceptible. So there are viruses that affect all different kinds of organisms. There are even viruses that infect bacteria themselves. Here are the top 12 deadliest viruses. Notice that we don't have COVID on this list, and that's because COVID is still relatively new and we're still learning a lot about it and how virulent it actually is. Other common viral diseases: hepatitis, viral meningitis, viral pneumonia, HPV, herpes, chickenpox, all caused by viruses. Viruses are not living. If you remember back to when we learned about the 8 characteristics of living things, viruses do not have the ability to reproduce on their own. So viruses are technically not considered a living organism. To reproduce, they must invade a host cell, and they basically hijack that host cell and cause that host cell to do their reproduction for them. So in terms of viral replication, a virus first attaches to a cell that it's infecting. It then will somehow inject its genetic material. Some viruses have DNA, but some viruses have RNA. When that genetic material is in the host cell, it takes over the host cell. It integrates with the host cell's DNA, and then as the host cell reads that DNA, it starts to produce new viruses. And then the new viruses are released from the host cell, and they're now able to invade other cells, and the original cell is killed in the process. So these are all examples of images that show viral replication. In terms of medications, antibiotics do not work on viruses. Remember, antibiotics are only effective for bacteria. There are different antiviral drugs, and these are prescription medications that somehow prevent the virus from multiplying. There are different drugs that interfere with different stages of the replication process. Some drugs prevent the virus from even attaching to a host cell. Some drugs will prevent the injection of the DNA. Some drugs will prevent the host cell from making viruses. So depending on the kind of drug and the virus that it's made to target, it will interfere with some sort of the stage of viral replication. Benefits of taking antiviral drugs is that oftentimes you'll suffer from a milder form of the illness. It can lessen the symptoms, shorten the time of illness, and reduce the risk of complications. Now, these medications are used in addition to our own natural way of fighting disease, and that's our immune system. The immune system is the body's defense against infectious organisms and other invaders or potentially damaging foreign bodies. There is specific immune responses that our body goes through. There are non-specific defenses, the first line of defense and the second line of defense. And then there are specific lines of defenses, which includes our third line of defense. So in terms of our first line of defense, this is non-specific. It's a protective barrier defense, and it prevents the entrance of any foreign body into our bodies. And some examples of our non-specific first line of defense would be skin, mucus, saliva, tears, sweat, sebum, cilia, good bacteria, stomach acid, and digestive enzymes. All of those are going to try to prevent something from entering our body. Now, if the first line of defense fails and we have a pathogen that has entered the body, then your body will kick into the second line of defense, which is still non-specific, meaning that this second line of defense can work against any sort of invader. So the second line of defense fights off what manages to enter the body. So if the first line of defense fails, we now have the second line of defense that's going to fight this off. One part of our second line of defense is the inflammatory response. Inflammatory response is when we see swelling. And this occurs because blood and lymph go to the infection site. And this can result in redness and swelling because we have that extra blood, that extra lymph going there. A fever often develops, and the way that a fever works is that the excess heat slows, stops, or even kills pathogens. So a fever is, in some ways, a sign that your immune system is actually working. And then phagocytes, which are types of white blood cells, can actually engulf pathogens. Now, if the first line of defense fails and the second line of defense fails, then your body needs to get more specific. And that's when it starts to organize the third line of defense. The third line of defense is when your body produces a very specific, unique response to the pathogen. So your body actually analyzes the pathogen that's infecting you and creates a special response just to fight that pathogen off. The body looks at the antigens that are on the pathogen. Antigens are markers on the surface of cells that are unique to each pathogen. So every pathogen would have different antigens or different markers on them. These antigens are recognized by our immune system as foreign or not belonging, and our body produces a different immune response to each different antigen. So there are a bunch of different cells that function in this third line of defense. So we have helper T cells, and they coordinate two different responses. One response that the helper T cells coordinate is called humoral immunity. And this is when we have pathogens in our blood and other body fluids. When we have pathogens in our blood and other body fluids, these helper T cells activate B cells. And the B cells will then turn into plasma cells. These plasma cells secrete antibodies that are very specific to only that one antigen. Antibodies will bind to the antigen that's on the pathogen. It neutralizes the pathogen. So when the antigen is bonded to by an antibody, it no longer can infect other cells. It also serves as a flag for those macrophages to come and engulf or eat and destroy that pathogen. So an antibody is a protein produced by the immune system in response to the presence of an antigen. Antibodies, proteins that bind to a specific antigen. Each antibody is unique and specific. Millions of antibodies respond to millions of foreign antigens. Antigens and antibodies have a lock and key relationship. Depending on the antigens present, specific antibodies are produced. The other kinds of cells that the B cells can differentiate into are memory B cells. These form and then remain in the body after infection. They're called memory cells because they remember the pathogen. So if you are ever infected with that pathogen again in the future, your immune system can result in faster antibody production upon that future infection. And so your body can produce all those plasma cells to fight off the infection before you even show symptoms of the infection itself. The other kind of immunity is cell-mediated immunity, and this responds to cells which have been infected with pathogens. In this case, helper T cells activate T cells, which differentiate into killer T cells. Killer T cells will kill infected cells, stopping the spread of the pathogen, and also memory T cells. These, like the memory B cells, remain in the body after infection. They remember the pathogen and result in faster immune response upon future infection. So memory cells in general are specialized cells that stay in the body after an infection is gone. And if the body is infected by the same pathogen again sometime in the future, the body can produce immune response much more quickly. So memory cells produce what's called the secondary immune response. This is the immune response triggered by memory cells. The fast response allows pathogens to be destroyed before symptoms even occur. Now, there's two different kinds of immunity. Acquired immunity can give you long-term protection from pathogens. Active immunity comes from production of antibodies and memory cells. It results in the secondary immune response, and it gives you immunity long-term, often for life. Natural active immunity is the production of memory cells from actual exposure to a pathogen. Artificial active immunity is the production of memory cells from artificial exposure to a pathogen. Artificial exposure to a pathogen could be through vaccination. In vaccination, an individual is given an injection of an inactive pathogen. This stimulates the primary immune response without the individual actually getting sick. And so it results in memory cells. Sometimes boosters are needed to make sure that that immune response and those memory cells are at full activity. The goal is herd immunity, which is also called community immunity. It's when a large part of the population is immune to a specific disease. Not every single person may be immune. However, the group as a whole has protection. With fewer infections, that actually does help protect those individuals who have not been vaccinated. So separate from active immunity is passive immunity. And passive immunity is from antibodies that are taken from another person or animal. And this gives temporary short-term immunity. Natural passive immunity is when maternal antibodies are passed on to the infant, either through the placenta during pregnancy or from breastfeeding after birth. Artificial passive immunity are antibodies that are injected from immune individuals. This is often obtained through something called convalescent plasma. This is from people who have recovered from disease, where their donated blood is centrifuged to keep just the plasma with the antibodies.